welcome everyone for the Institute lecture today by Professor Eric Green of Columbia University. I have known Eric since almost exactly 20 years from now, in almost 20 years. And he was an assistant professor at that point of time at the Columbia University in the medical school in the Department of Biochemistry and Molecular Biophysics. Before that, he was a postdoc at NIH, and prior to that, he got his PhD from Texas A&M in biochemistry. And what is most important, he has got many awards and accolades. I don't want to get into the list of many of those things that are there, but what I think he is most well known for, and even here in India, many people have asked me the question related to something that he developed, was the DNA curtains, this assay or the technology of DNA-based curtains, with which the people in the world, first Eric's group and then the rest of the world, could study interactions of proteins and DNAs one at a time, so one-to-one reactions, and you can look at thousands of such reactions at single molecule level. And that development of that technology opened up a completely new field and allowed scientists to look at a wide variety of biological processes which involved interactions of DNAs and proteins. So I don't again want to say too much about it. I think his seminar itself, he has been here in IIT Bombay 10 years back or maybe 12 years back. He gave a talk in our department, in the Department of Chemistry, and I remember that room was completely full and people were standing. So a colleague of mine suggested that we should have an institute lecture because his field of work cuts across different disciplines, starting from physics, chemistry, biochemistry, a bit of engineering as well. A lot of technology development has gone in. So maybe we can learn more about some of the stuff from Eric. Eric, the floor is yours.
Thank you, Arindam. That was very kind. And thank you all for attending my lecture, and hopefully it's reasonably interesting. My lab mostly works on DNA recombination, so a lot of the talk is going to be about recombination, and I'll give you an introduction to that field at a very simple level so that we all understand it. And I'll also talk about the technologies that we have developed to study DNA recombination, much of which we were developing at the time that Arindam was in the lab. And then I'm going to talk about something that we worked on for a really long time. And in fact, we started working on it before Arindam came to the lab, and it only took us 17 years to actually make it work. And so I thought maybe Arindam would remember this problem. Maybe he doesn't. It's actually the problem that I presented to the faculty in my department when they were seeking new hires, and I said, hey, we're gonna solve this problem. So hopefully it's interesting. And the title of my talk is Single Molecule Studies of Homologous Recombination, just because that's the most generic title that I can give you, okay? And so I want to highlight the folks who've done most of the work. Like Arindam, we just sit behind a desk and look kind of cranky and say stuff. It's really the people in the group who will have done everything. And the person who is responsible for almost everything I'm going to tell you about today in terms of the science is going to be a guy named Brooks Crickard, a former postdoc of mine. I'll show you a picture of him in just a moment. These are my current people. And I want to point out, I'm currently hiring postdocs. And just so you know, I'm going to talk a lot about single molecule stuff, but we also do a lot of work now in cryo-EM, a lot of work in genetics. I'm always happy to talk to people in theory, anyone who's interested in the types of problems that we think about. So again, I'm not the person who did the work. It was my former postdoc, Brooks, who's now an assistant professor at Cornell in Ithaca, New York. So he's actually doing incredibly well out on his own. So this will be an outline of everything that I'm going to tell you. I'm going to start with very brief introduction to homologous DNA recombination. I'm going to talk about the homology search problem. This is essentially a question of how do two biomolecules find each other in a cell, and in this case, it's how do two DNA sequences that are homologous to one another find each other. I'm going to give you an introduction to that problem. I'm going to talk about DNA curtains so you know the methodology that we used and how we've developed it and some other things that we use it for. And then I'm going to get to this last part of the talk, which will be the bulk of the experimental work that I want you to understand. It's how does this protein RAD54, which I'll tell you about as we go along, contribute to this homology search problem. Okay? This is my simplified biophysics view of DNA recombination. I want you to look at this with a caveat of understanding that in us, in humans, there's about 40 proteins that are involved in this process. I'm only going to talk about a couple of these, but if there are questions about others, we can discuss that at the end. And so this process, as I have outlined it, occurs in bacteriophage, it occurs in bacteria, it occurs in yeast, it occurs in humans, and this process is essentially the same thing is going on, although with slightly different sets of proteins. So what happens is you get a double-stranded DNA break. These are the most lethal type of DNA damage that you can incur. So if you get one of these breaks, the cell will either die, fix it, or become cancerous. That break is then processed through this end resection reaction to generate long single-stranded DNA 3' overhangs. And that overhang serves as the platform for the assembly of a protein in humans called RAD51. In bacteria, it's called REC-A. These proteins make long filamentous structures on that single-stranded DNA, and that's going to be called a presynaptic complex. And I'll be coming back to that complex as I go along with the talk. What that complex then has to do is search other regions of the genome, depicted here in red, until it finds a sequence that is complementary to that 3' overhang. It then has to drive invasion of that black DNA strand into the red DNA molecule, displacing part of that strand. And this displaced strand, or this whole structure, is called a D-loop, or a displacement loop. What then happens is that 3' end can serve as the primer to initiate new DNA synthesis. And so you're generating new DNA, essentially replacing the information that was lost during this breakage and processing step. And then this intermediate can be disrupted in a number of different related mechanisms, which I'm not going to go into because they're quite complicated. The most simple one is it simply gets unwound, and this newly synthesized DNA pairs with this overhang, and the gaps get filled in. But ultimately, the idea is that you regenerate the continuity of your originally damaged DNA strand. Okay? So now my lab works on how do proteins bind to this end, how do those ends, how is that process regulated, upregulated or downregulated, how are proteins involved in cancer involved in this process, and how does this end find the homologous template on the red DNA in this case. And just so you know, in terms of background, this is crucial for genome integrity. If you knock this process out, for example, in yeast, and you give them one double-stranded DNA break, they die. A lot of cancer drugs are intended to disrupt these types of repair mechanisms because cancer cells have to replicate so much faster than other cells. If you disrupt their ability to repair DNA during that replication, they can die. So this is directly related to cancer biology, and many of you have probably heard of the proteins BRCA1 and BRCA2. So these are hereditary proteins involved in hereditary breast and ovarian cancers. They're directly involved, actually, in allowing this step to occur, the assembly step. There are also cancer-prone syndromes. So there's a syndrome called Bloom syndrome, where the diagnostic for that disease is they simply look at your chromosomes. And the chromosomes are so disastrously messed up that they can tell just by looking at them that you have this major problem. And the phenotypic consequence for the patient is that they're essentially dying in their early or mid-teens to late 20s of cancer. So they're very cancer prone. There are actually a number of diseases along these lines. And I talk about this in terms of DNA damage, DNA damage can come in the forms of chemical damage, ionizing radiation, cancer drugs that intentionally do this. The other thing that causes damage is replication errors. Anytime you have a replication fork that hits something that it can't deal with, you can end up with this type of damage or something closely related. And you need recombination to restore those replication forks. And in fact, that's probably where it's most active in normal cells that are undergoing division. And of course, anytime you're talking about genome engineering, almost always with a CRISPR-based mechanism, for example, that's driven by horizontal gene transfer, which is recombination. And then it's also deeply involved in meiosis and generation of genetic diversity. So if you look at your mom and your dad, you might look like one or the other of them. You might look like your siblings. Very rarely do you look identical to them. And the reason for that is you have genetic diversity generated by the recombination of genes that are coming from your mother and your father, so that you're not exactly like either of those individuals. So now, I'm going to be talking about the black DNA finding the red DNA. And this is what we call the homology search problem. So essentially, how are two different DNA molecules aligned with one another during this recombination process? And the importance of that is if you misalign, you actually drive recombination with the wrong sequence, and you get what are called chromosome translocations, which can directly lead to cancer. And so you're taking this Rad51 filament and you're searching the DNA, and the question is, how do you actually end up in the right spot so that this pairing reaction can occur properly? Now, there's been a handful of studies done on this. Work from my colleague Steve Kowalczykowski, our laboratory, Takjeep Ha's lab. Ultimately, these studies all suggested that the processes were driven by various forms of diffusion. So molecules essentially randomly searching until they find one another and can align. And the mechanism can occur either through what's called intersegmental transfer, where you have a long polymer, part of which binds to your red DNA, another part binds somewhere else, and it sort of walks along in this hand-over-hand manner, if you want to think of it that way, or random three-dimensional diffusion, or one-dimensional diffusion. So it looks like all of these diffusive-based processes can contribute to this reaction in a test tube with a minimal set of reaction factors. And so in each of these articles, ourselves, or Steve, or Takjeep, we were studying just the recombinase, either just REC-A or just Rad51. And so we came up with these ideas of diffusion-driven alignment of the sequences, okay? So now, I'm going to change gears a little bit. I'm going to talk about DNA curtains, the technology that we developed. Then we're going to come back to the target search problem and discuss how this new accessory protein, Rad54, contributes to this. And so we're going to take the studies that we've done and now add an important accessory factor, okay? So, DNA curtains as tools. This is something that we developed sort of in the early days of my laboratory, around the time that Arindam was there, maybe just a little bit before. And we've used it quite successfully ever since. In fact, we're launching a company right now to sell these things. So let me explain how this works. And first, I want to preface that by saying, why did we develop this? So when I was a postdoc, the way that I would do single molecule measurements is I would put a DNA molecule on the surface of a slide, and it would just be randomly anchored. So we would put biotin on the surface. There'd be a small fraction of biotin that had streptavidin on it, and then we would stick a biotin-elated DNA on there. And so you'd get these sort of random distributions of molecules. And so what that meant for me, sort of in a practical sense as a postdoc, is I would spend an 8-hour day in a very small room all by myself collecting data. And at the end of the day, on a good day, I would have data from 5 molecules. So it was extremely difficult. Okay, so that was problem number 1, the low data throughput. The second problem is perhaps a little bit harder to understand unless you've done single molecule work, but I'll try to explain it. So we're working within the context of a microfluidic sample chamber, yet we're trying to do biology. And so we want those molecules in that chamber to behave in a way that is as normal as possible given the experimental constraints. And so now, if you think about just a glass chamber, if you inject protein into that chamber, the first thing that's going to happen is that protein is just going to glob onto the surface. It's going to absorb non-specifically to the surface. And because of that, it's not doing anything that's biologically relevant. And so what we needed was and let me illustrate this with a fun anecdote. As a postdoc, I studied two proteins. One was called mu A and the other was called mu B. They're DNA transposition proteins. Their details don't matter. After we built a microscope, after we developed methods for making flow cells, figuring out how to put DNA on the surface, it took me 6 months to find conditions to keep mu B from just sticking on the surface. And essentially that was through tweaking buffer conditions, tweaking the pH, and adding a protein called alpha casein, just a random protein that would absorb to the surface. And so I spent all this time getting mu B to work perfectly, and then I add its partner protein, mu A, and lo and behold, it goes in and sticks to the surface. And again, we're back to square one, and it took another 6 months to figure out conditions under which both proteins worked. And so thinking ahead to my own career, I knew I wanted to be working on much more complicated systems where the molecules themselves, there would be lots of them. And I realized, okay, if I have to adjust conditions for every different molecule I'm putting in here, I will not be able to study that much stuff. And so we really needed a generic way of passivating a surface that would work with as many things as we wanted. And so the strategy was to take now our microfluidic surface, and instead of coating it with BSA or alpha casing or some random protein, we instead put a fluid lipid bilayer on the surface. And the logic there was fairly simple. Inside of cells, proteins are always surrounded by bilayers, and so we thought this should provide an inert environment where proteins will not just absorb to a surface. So that's point number one. The second issue is we knew, okay, if we put the DNA on the bilayer, what's going to happen? And so the idea was, again, we have surface coated with bilayer. We now have a subset of lipids within that bilayer that have a biotin on them. So those lipids are diffusing freely in 2 dimensions. We can then stick a streptavidin molecule there, and we can stick our DNA on that molecule. So this is now our DNA diffusing in 2 dimensions on the surface of a microfluidic sample chamber. And we realized if we place a hydrodynamic force on that molecule, what's going to happen is we're going to flop that molecule over and we're going to push it in the direction of the hydrodynamic flow. And then we very quickly realized that we would be able to use that as a tool for controlling the DNA. If we could control the distribution of the lipids on the surface, we could control the distribution of the DNA. And so that's where we began to think about barriers to lipid diffusion. And the idea is very simple. It had been known for a number of years from the work from a chemist called Steve Boxer that if you put metallic patterns on surfaces, lipids would not traverse those patterns. And so what we did was take our microfluidic surface chamber, we engineered a small piece of metal onto that surface using electron beam lithography. We actually have multiple pieces of metal. Now we take our DNA attached to the bilayer, diffusing in 2 dimensions, and apply a flow force. And that DNA is going to hit that barrier to lipid diffusion, that little tiny piece of metal. It's going to fold over, and we're going to be able to look at it by something called total internal reflection fluorescence microscopy. This is essentially a microscopy technique where we're just illuminating a very thin sliver. So now, barrier, I'm going to take that slide, I'm going to turn it towards you and rotate it. Here's our DNA. Here's our barrier. We're going to push them in, and they're all going to line up along the leading edge of that barrier. And so instead of, in principle, looking at just a few molecules, we now be able to look at perhaps hundreds of molecules all aligned with one another. So now, this is what I just told you in words. Here's our microfluidic sample chamber. Here's our lipid-coated surface. Here is a barrier to lipid diffusion made out of metal, in this case chromium. We make it a zigzag pattern because the zigs and zags can control the distance between the DNA. It's a minor point, we can talk about it later if you're interested. And then these are all the DNA molecules that we've pushed into that barrier and flipped over. And so what I'm going to do now is show a video actually done by a student who was in my lab when Arindam was there, a wonderful woman named Marilyse Visnapu, who would probably laugh if she knew I still showed this video. So this is what I would call a 4-tiered DNA curtain. There's a piece of metal here, a piece of metal here, a piece of metal here, and a piece of metal here. And what we've done is pushed all of these DNA molecules into that metal. And so what you're looking at, when it stretches out, all of the green, each of those green lines that you see is a single molecule of DNA. In this case, it's 50 kilobases in length. All of the purple spots that you see are actually individual fluorescently tagged nucleosomes. And what we're doing in this video is we're pushing the DNA so that it's parallel to the surface and we can see it with this special illumination technique. And then we're transiently stopping flow so that the molecules diffuse up away from the surface. And so this was early days in the development of the technology, and that was the tool that we used to show the reviewers that the proteins weren't just stuck to the surface, but they were actually bound to the DNA molecules and doing what we hoped was biology. So now, the proteins that you see here are actually eukaryotic nucleosomes that we've deposited onto the DNA by salt dialysis. And the fun part of this technique, well, the first thing is that they're not sticking to the surface, maybe a few, but they don't matter. Instead of 3 to 5 DNA molecules, I had Marilyse sit down and count these. There are 805 DNA molecules in this field of view. Each one has about 10 proteins bound to them. So now we're talking about thousands and thousands of individual protein DNA interactions that we can look at. So even folks that have had no experience with single molecule data can look at this video without any detailed analysis and immediately tell a couple of important things about how this protein is behaving. So the first thing is the protein distribution on the DNA looks completely random to your eye, okay? I'm going to give you a counterpoint example of this in just a moment. The second thing is the protein is not moving. It's staying stationary on the DNA within our resolution limits. The third thing is it's not falling off the DNA. So immediately we have information on binding site distributions, lifetimes, and movement behaviors, okay? So keep that in mind. This is a counterpoint to that. So in this case, you're again looking at a 4-tiered DNA curtain. The DNA is labeled in green. The protein is labeled in purplish. This is exactly the same protein. These are eukaryotic nucleosomes that we've deposited on the DNA by salt dialysis. But everybody in this room can say, hey, wait a minute. Now it's binding to specific sites on the DNA. And so the difference with this DNA molecule is we've gone in and engineered into it something called a Widom 601 sequence. This is an artificially selected sequence developed by John Widom way back when that binds very, very tightly to nucleosomes. And so this is essentially acting as a sink to suck up all the nucleosomes and drive them into single positions. And so we can make these types of measurements just by eye, saying, yeah, we know exactly where this protein is binding. And we, of course, go in and do detailed analysis of that. But from these images, we immediately have a sense of what is the protein doing. So now, I want to talk about a little bit of a different flavor of DNA curtains. 50 kilobases. All of those are 50 kilobases, yeah. So now, imagine you are a graduate student and your advisor, Arindam, says, hey, or Ruchi tells you, to, hey, go purify this protein, and it takes you weeks and months and however long to learn how to do it. You've got all this material, it's in your tube. In our system, what you do with that tube is you inject it, it goes through the sample chamber, and then it goes out of the sample chamber into the trash. And that is because we have to have constant buffer flow to see the molecules, otherwise they diffuse up out of this evanescent field, of the turf field that we're looking at, and we can't see them. So we waste a lot of material. And so we also wanted to develop methods that we could look at proteins where we don't have a perturbing flow force. And so in this case, what we did, this was actually done by a combination of students all of whom Arindam knows, Jason Gorman, Marylise Visnapu, and Teresa Fazio, they developed a way of anchoring the second end of the DNA to these pentagonal anchor points. Essentially what we're doing is pushing the DNA into the barrier at the front. It flops over, and those free ends of the molecules have a hapten, typically a digoxygenin, and we put antibodies on these pedestals that can then grab onto the DNA. So this was something that we developed that made me very, very happy. This is the first example. I think Jason collected this data. There are about 100-something DNAs here, but all of these linear lines that you see are individual strands of DNA. There is no buffer flowing here. And so this was a big breakthrough for the lab, okay? Now, I'm gonna show you a video, this same type of curtain, but now with proteins bound. This was, again, data collected by a former student, Jason. So this is a protein called MLH1 PMS1. Details aren't important. What it does is it searches DNA for a specific type of damage. And we wanted to know, okay, how is it searching the DNA? We had very little understanding at that point of how that protein searched DNA. In fact, we had very little understanding of how other proteins search DNA. And so you can look at this, and immediately it looks very, very different from what I showed you for the nucleosomes. The first thing is that all these little spots are moving, right? And so we can go in and analyze that data and show that this is pure one-dimensional diffusion. So the proteins are essentially binding to the DNA and undergoing a random walk along that DNA. And although I don't show it here, when they encounter the damage that they're supposed to interact with, they walk around, and stop right at that damage site. So we know that this guy searches DNA. And in fact, it goes right over nucleosomes. So in this particular instance, what you're looking at are fluorescent quantum dots, which at that time in these cases, they actually varied somewhat in intensity. We didn't really know the molecular organization of this molecule, and there is some sense that it can form larger oligomers. I think most of them in this case are probably not oligomers, but there may be some. This is an interesting protein. It forms a great big ring, 50 nanometers diameter. We think it's binding to the DNA on the inner surface and hopping along the DNA from that inner surface, and that inner surface is much larger than the diameter of a nucleosome, so it's essentially hopping along the DNA. Yeah, absolutely, that's what we think it's doing. So when we add free DNA to this one specifically, it does not compete off of the DNA. But if we take that molecule and we cut it, and then we add flow so that it flips over, that protein pushes right off the end of the molecule. Yeah. Everything I've talked about so far has been double-stranded DNA. Everything I'm going to talk about now is single-stranded DNA. And so this was another huge breakthrough for the lab because a lot of what we study, we try to mimic that single-stranded overhang that's generated during recombination. And so we really spent a long time trying to figure out how to make single-stranded DNA curtains. And the simple trick was we take a circular single-stranded DNA, we have a little primer that we anneal to it, and we do something called rolling circle replication, where we have a polymerase that will just go round and round and round and round and make really long stretches of single-stranded DNA, and then we bind them by something called RPA, replication protein A, which is present in all eukaryotes. And it will remove any secondary structure. And in this case, it's GFP tags, so we can also label the DNA. And so what you can see here is we can generate the DNA. Here it's already made, but it's getting longer and longer and longer by virtue of the binding of this RPA protein. And we think that this guy will actually mimic this overhang. And so we use this for a tremendous number of studies. All right, so now, for the rest of the talk, I want to get back to this homology search problem. And this protein RAD54. And remember, this problem is basically how does your black DNA find your red DNA and not get in the wrong place? So RAD54 works together with RAD51 during recombination. RAD54 is what's called an SNF2-like protein. So it's an ATP-dependent protein. It's a motor protein. It's present in all eukaryotes. We have it. In fact, we have two different kinds. Yeast have it. All this work I'm going to talk about will be with yeast proteins that are purified. It is absent from bacteria, which if I remember this, I'm going to come back to that point at the end. It is a double-stranded DNA-dependent ATP that is stimulated by Rad51 only when Rad51 is bound to a single-stranded DNA molecule. It is a motor protein. It physically interacts with Rad51. It's known to remove Rad51 from double-stranded DNA. And it's thought to do all kinds of things like take apart bad recombination intermediates, remodel chromatin. We know it goes to DNA breaks. It causes the chromosomes to move around, or at least contributes to that. We don't really understand that process. And it's absolutely critical for eukaryotic recombination. So if you take yeast, you knock Rad51 out, the recombinase protein itself, you induce one double-stranded break, the yeast dies. If you take yeast, you knock Rad54 out, you induce one double-stranded break, the cell dies. And so we know that the presence of Rad54 in yeast is just as important as the presence of the recombinase itself. Okay, so the question is, what is it doing? So what I want to do is just try to outline the reaction for you as we know it in terms of the GFP tag protein that we've made. So we do try to be careful to make sure that the fluorescent proteins we're making function in a way that is proper. And so what Brooks did was generate GFP Rad54, expressed it in yeast, purified it. We know that if we put it into yeast, our colleague Rodney Rothstein showed a number of years ago that the yeast are fine. The protein goes to breaks, repairs the breaks. There is no problem biologically. What we now know is if we take GFP Rad54, remember I told you that it was stimulated by Rad51. If we have an ATPase reaction without Rad51 versus with Rad51, we see a strong stimulatory effect. So we know that it's responding to Rad51 correctly. And in fact, we can take unlabeled Rad54 or GFP tag Rad54, and we get the same level of stimulation. And so we know that it's behaving properly in a test tube. I'm going to explain the assay that we use to look at essentially how does the red DNA and the black DNA become paired in a test tube. It'll be important to understand this assay because this is what we use to derive all the single molecule data that I'm going to show you. And so in this case, what we're doing is taking a small DNA fragment intended to mimic that processed DNA overhang. Here we have a 56 base pair duplex DNA and a 21 nucleotide overhang. We have a fluorophore so that we can see it on a gel. And we're adding a bunch of components. We're adding replication protein A. We're adding ATP. All of this stuff is ATP dependent. We're adding unlabeled Rad51. We're adding GFP Rad54 or unlabeled Rad54. And we're making this complex that I mentioned earlier, the presynaptic complex. So this is the thing that forms on that DNA overhang and is trying to find where do I pair it with so that we can fix the damage. And we mix this with a plasmid, we incubate, we remove all the proteins, and we run a gel. And the question is, can it form what I mentioned earlier, a D loop, where the black DNA and the red DNA are paired? And so this is a gel showing a reaction in the absence of Rad54. You get nothing. If we put in wild type unlabeled Rad54, these are the reaction products after 10 and 30 minutes. If we put in GFP Rad54, this is 10 and 30 minutes. And so we know the protein works, and we can quantify this and show that the unlabeled and the labeled protein are essentially working the same. This has been characterized for years and years and years. You can actually go in and cut that displaced strand with single-stranded specific nucleases, for example. Yeah, so you can design that overhang toward any DNA molecule you want. If it's random and not targeted to PUC19, you actually don't form that intermediate. Is that, yeah. All right, so now, the key here was taking this exact same reaction and applying it to an experimental format where we can now try to see how did that DNA molecule find the right target. And so what Brooks did was take a DNA molecule that he derived from PUC19. He essentially took PUC19, put a big stuffer in it, cut the stuffer in half so the PUC19 is in the middle, and then he tethered it between a barrier and anchor points. And the idea was to inject this fluorescently tagged complex and ask, can we see interactions with these double tethered DNA molecules? All right, so I realize I should have explained something earlier, and I have to explain it now. So when we're analyzing these data, typically I present it either as a movie or as a chymograph. And so what I'm going to show you now are chymographs illustrating how these molecules change with time. What I'm doing is taking a single 1-pixel slice. I shouldn't say what I'm doing. What Brooks was doing, what Arindam did, was take a single 1-pixel slice through one DNA molecule and then project that with time, okay? And so that's shown here. And I want to explain chymograph a little bit more before I talk about the data. So this is one DNA projected with time. This is distance. This is time. So here's our scale bar. So if we take these molecules and look at them with GFP-Rad54, what we would see is, okay, you get a binding event, and then all of a sudden, that molecule would start moving. And we can follow this by following either the GFP-Rad54 or the ADO-labeled DNA fragment. So we can see pink or magenta versus green. So we know that Rad54 is bound to that little DNA fragment as we've designed it, and we know that it can move that DNA along the other DNA molecule now. Hopefully that's not too confusing. Essentially what's happening is we have a tethered double-stranded DNA. We have a little presynaptic complex coming in and binding to it, and what it looks like is that all of a sudden it starts moving, okay? So now we have a mutant of Rad54 called K341R. This mutant binds DNA just fine. It does everything just fine. What it cannot do is hydrolyze ATP. And so when we put that guy in, all of a sudden we see binding, but we see no evidence of this type of movement. So this immediately made us think, okay, somehow Rad54 is acting as a motor that's driving the motion of this little DNA fragment along a much longer double-stranded DNA. And just a couple of quick controls. So if we don't include Rad54, we actually don't see any binding. This is our red channel. We don't see our little DNA fragment. This is our green channel. What we're doing is labeling the double-stranded DNA at the end with a dye called yo-yo-1 just to show that it's there. We don't see any binding. If we omit Rad51, again, we don't see any binding. So we know that we have to have both proteins of the presynaptic complex there. And it's only when we have both do we see binding and this type of movement. Now, the movement itself can be characterized just in terms of the translocation characteristics. So we're seeing velocities on the order of 150 base pairs per second. They typically go a distance of about 5 kilobases before they turn around. We know, and we do not understand this, that this 150 value comes from a 21 nucleotide overhang, as I showed you in the schematic. We can actually make that overhang longer and longer and longer, and it will keep going faster and faster and faster up to about 800 base pairs per second. We do not understand this length dependence, even after a couple of years now. We know that, I don't want to belabor this, that the wild type versus the GFP behaves the same in terms of velocity, in terms of the distance. We can do Gaussians of both, and they come out roughly the same. So we know now this thing is moving the protein along the DNA, the protein complex along the DNA. The question is, does it actually recognize the target? If it doesn't recognize the target, then we've got a problem. And so what Brooks did was generate 2 sets of DNA molecules, one that had a target here at what he called the 4.5 position, or here at the 8.6. If we target the 8.6, we get a buildup of complexes that hit 8.6 and just stop. We target 4.5, they hit 4.5 and stop. This is all with GFP tag Rad54. If he does the same thing with unlabeled Rad54, he gets the same type of binding. And so we can see peaks in the binding distribution after 10-minute incubation corresponding to the target site. We can actually do something very interesting at this point. So we know where the targets are. We can see movement. We can see that it gets to those targets by moving in one dimension. And we can ask, what is the first passage recognition efficiency for the target search? This is actually a question that I think is of dramatic interest to people doing, for example, how do transcription factors find targets? How many times do they have to hit the target before they recognize it? As far as I'm aware, we're the first people to measure this for any type of complex. And so here's our 8.6 target. Here's a guy that happens to go right on past it and doesn't see it. Here's one that goes to it and then just stops dead in its tracks. About 30% of the ones that we see stop and bind, and the rest simply bypass it on the first event. And this is true for 21, 90, 150, and 1,000 nucleotide single-stranded DNA fragments. And so that 30% value seems to be the upper limit for the target recognition efficiency in this assay. Now, I want to talk about the homology recognition requirements. So we can actually see recognition for even these smaller 7, 9, and 15 nucleotide overhangs. We don't see these intermediates in gel-based assays because they're too unstable and we think they fall apart. So 21 is the first place that we can actually map intermediates in a gel assay. So I've mentioned now 30% recognition efficiency. Another interesting facet of these things is they don't always go straight. Oftentimes they can turn around, and they can turn around multiple times. In fact, we can plot the probability versus the number of reversals, and we can see, hey, if they miss the target, they can actually come back and hit it again, okay? Now, this is going to be complicated to explain, but I'm going to try because it's super interesting. So we know the sequence of the DNA that we're putting in. We know the sequence of the DNA and its orientation that we have tethered. We know in what direction this thing should be coming along to hit the target. And we also know that if it comes in the other direction, it should not hit the target because DNA is anti-parallel. It should only be aligned in the correct orientation from one direction. This is the way that I thought about this. So if you think about RNA polymerase and RNA aligned with a DNA within the active site of polymerase, you can only bind to one strand. You can't simply flip around and bind to the other strand because that RNA is only complementary to one strand. And so that's the way that I thought about this. I thought, okay, it should only recognize the target coming from what we call the correct direction. It should not recognize it coming from the incorrect. And so this is going from A to B, what we call the incorrect direction, yet it stops right where the target is. This is coming from B to A, which is what we call the correct direction, and yet it stops right where the target is. And so somehow it doesn't care which strand the target strand is on, okay? And in fact, we can show that this actually varies with the length of that overhang. And so this is the correct direction, this is the incorrect direction. This is the correct direction, this is the incorrect direction. For short nucleotides, short nucleotide overhangs, it's about 50-50. When we go to a longer direction, it actually has a harder time going in the incorrect direction, but it still does it quite well, okay? Now, I can plot that data. I don't want to belabor this, but again, you can see correct direction, incorrect direction varies with length. The interesting thing about this is I mentioned earlier that there's a variation in velocity with length. And so if you think about this in another way, incorrect direction recognition becomes more difficult as you're going faster, okay? So we think that slowing everything down helps it to recognize things from both sides. So now, I mentioned this protein RPA, replication protein A. This is in our reactions, but we didn't know what it was doing. We knew it was required. If you omit RPA, you get some D-loop, but not nearly as much as if you have RPA. We know that we can remove RPA and we still see this movement, we still see the same velocities, they still go the same distances. So why do we have to have RPA for the reaction to work? We also knew, or we found out, that if we labeled the RPA and labeled our little DNA fragment, that the RPA actually bound to the fragment after some delay and then tracked with it as it moved along the DNA. And so this was the first indication that RPA itself was doing something within the context of the presynaptic complex as it was searching the DNA molecule, okay? So we know that it has to, we know that it's there, we know it co-localizes with Rad54. If we block these reactions with mutants of Rad54 that can't hydrolyze ATP, or with non-hydrolyzable ATP analogs, we actually block the association of RPA with those molecules. So we think RPA binding to these complexes has something to do with those complexes hydrolyzing ATP and presumably moving along the DNA. We can also quantitate the amount of RPA. I won't belabor this, but it's about, we think, 3 molecules of RPA per presynaptic complex, somewhat independent of DNA length. Now, we know that in the absence of all these fun little DNA molecules and everything, if we just put Rad54 itself into these reactions, it will bind and it will translocate. We know that it doesn't depend upon RPA in terms of anything it does. This was published many years ago by my colleague Steve Kowalczykowski. But we can also show now that RPA, in this case RPA-M cherry, will co-localize with Rad54 as it translocates along that molecule and will follow it along the DNA. So now, we know that if we block that translocation with a mutation, we don't see co-localization. This is showing co-localization, this is showing none. We also know that that co-localization isn't really dependent upon any protein-protein interactions that we can find. So RPA does not interact with Rad54 in and of itself, and we can see co-localization of human RPA with yeast Rad54 as well as E. coli SSB with yeast Rad54 searching the DNA. So we think there's something here to do with single-stranded DNA as a general characteristic and nothing to do with any protein-protein interactions. I'm going to come back to this. And so the question is, what is RPA doing? We know in a test tube you have to have it there, but in the reaction, it doesn't seem to have any effect on the motion that's taking place. And so what we did was look at target recognition efficiency. That is, when you're coming along and you hit the target, how often do you stop? This is 21, 90, 150, 1,000 nucleotide single-stranded overhang. These are plus RPA and minus RPA. And so we can see that there's a modest difference in initial passage recognition efficiency when we have RPA versus not RPA. So we can see that there's a drop from about 35% or 30-ish% down to about 20-ish% for each of those. And so now what Brooks did was take all of that data and subdivide it into correct direction versus incorrect direction. And so these are the black data sets showing the correct direction. These are the incorrect direction for each of the different lengths. And so if we have our 21 nucleotide guy, it's about a 50-50 split as long as we have RPA there. As soon as we get rid of RPA, we start favoring the correct direction. And this bias occurs all the way out to these much longer substrates where when you have RPA versus not having RPA, you get a bigger difference in terms of overall recognition in the what I call the correct direction. Now, that gets us to a model. And I want to preface this by saying this is a model. And it's a model built upon a reductionist biochemist view of what's taking place. We can talk about that as we go along. We do not have structures of anything here. So we're hoping to generate these as we go along with the lab. So what we think takes place is we have our presynaptic complex shown here with Rad54 bound to it. We think it's actually binding to this double-stranded DNA fragment, and we think it's opening that fragment. We think that opening is double-stranded, dependent upon the ATP hydrolysis activity of Rad54. At that point, we think RPA molecules bind to that opened DNA strand. So in a sense, it's unwinding the DNA and giving RPA a place to bind. This then translocates along the DNA while hydrolyzing ATP until it hits its target. And somehow you hit that target and you go from 150 base pairs or whatever you happen to be translocating at to 0. You stop dead in your tracks when you hit the right target. We do not understand that. And so how then does this come out to this idea of correct versus non-correct target recognition? And so what we think might be happening is this is a simple drawing where you have the 3' end scanning along this DNA strand, and this is the target strand. Okay, so that means this is 3', this is 5' to 3'. That's easy to understand. What we think might be happening is there are transient bends in this filament or bends in this open bubble itself that allow now these strands to show opposing polarities, even though they're in what I call the incorrect direction. And so the fun thing about RPA is that the first structure, which was solved by Nikola Pavlitic, shows that RPA bends DNA by 180 degrees. And so we think RPA might actually be binding to this, bending the DNA around, so now that you have correct polarity no matter which of the two strands that you're probing. And so the important part of this is that You bind your chromosome, you start searching for red DNA that matches you. If you can only recognize it from one direction, you're going to miss it 50% of the time. Because now you can scan both strands, if you hit your target, you have a much higher, presumably a higher likelihood of stopping at the right place. And so now I'm going to hit my summary slide. I've talked about DNA curtains as a fun tool. What I've shown you, if you don't remember anything else, is that Rad54 is a very active participant in this process. If you don't have Rad54, you have a diffusion-driven process. With Rad54, you have a motor-driven process. We show that RPA co-localizes with search intermediates, and we think that Rad54 is unwinding that DNA and allowing RPA to do that. Now, I mentioned very quickly early on, E. coli bacteria have no Rad54. There is no equivalent protein to Rad54. And so we think that in bacteria, this process may be fundamentally different and driven primarily by diffusion, whereas in eukaryotes, it's driven primarily by these motored protein-driven mechanisms. Thank you all for your attention.
Thanks, Eric, for this lovely, lovely talk. Let's first have questions.
Very nice, super talk. And I have a question about this correct versus incorrect part.
Yes.
So, is it possible that the double-strand DNA, some of the part could unwind and that would rotate the orientation? Is it?
So I think it is unwinding, and I think that unwinding is driven by Rad54. But remember that correct versus incorrect is really influenced by the presence of RPA. And so that's why I think RPA is doing something to the DNA molecule itself that's influencing recognition in the incorrect direction. But in essence, it's being driven by the underwinding of the DNA. Yeah.
And how processive is this motion?
So typically what we're seeing is that it goes about 5 kilobases before then it turns around. So it can go quite long distance cumulatively, but it's on it's a handful, 5 kb in one direction and then going back the other direction.
It doesn't fall off.
We don't see it falling off, no. Well, we do, but not mostly after tens of kb. Yeah.
So I have a related question.
I want to add a caveat to that. Again, reductionist experiment. Whether it's falling off in a cell, I assume it has to fall off relatively frequently, especially if it's in the wrong place, because then it needs to get to some right place.
So I have actually a question related to this turning around that you showed. It's clear that it's going back and forth, but I mean, how does it, I mean, if it's a motor protein, right? I mean, is it coming off and then going around and then reattaching itself, or is it some other inherent mechanism that it's, is there any idea on that?
We actually think it's going, partially dissociating, and then it's just randomly doing something, and sometimes it lands and goes the other direction.
Yeah,
this is an idea that we and others in the field have had for not just this protein, but others. I had a postdoc named Jail who worked on another protein called FTSK. This is a protein that sits at the division site between two bacteria. And it makes a ring, like a helicase-like ring that drives DNA from mother to daughter cell, particularly in Bacillus subtilis. So it wraps around DNA. We can put that on here. It's super fast, like 30,000 kilobases per second. It's crazy, just ding, 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 ding. Highly processive. It turns around very, very frequently. And so you have a complete ring that's somehow coming apart, flipping around. And in that case, we have a structure. We know that it can only go in one direction based upon its orientation on the DNA. But in that case, it's totally coming apart, going back on, and just taking off in the other direction. So that's exactly what we think is happening here. We would love to have, proof of that. I don't say we have proof, but that's our model for what's happening. Okay. Yeah. So we do think that Rad54 has to bind correctly to Rad51 for the whole thing to work.
Basal activity, I saw that it had basal ATPase activity in the absence of Rad51. So it was working. It was just not working efficiently.
So Rad54, if you give it double-stranded DNA, it will hydrolyze ATP. It's this high stimulation you get when you have Rad51 around.
So does it?
Rad54 itself without anything, is a motor protein.
So
for
I was
sure.
just thinking, is the translocation got anything to do with RAD51 binding so that it goes in one direction for a while and then maybe the complex dissociates and then it can turn?
I suspect that it's a combination of RAD51. So if you think about RAD51, it itself has to be polar because it's bound to a polar single-stranded DNA. And then RAD54 has to bind to that. And so the entire polarity of the presynaptic complex is going to be driven by the orientation of the single-stranded DNA fragment to which it is bound. Okay, and so that's what I think is driving the initial interaction, the initial polarity. I don't think the whole complex is coming apart. The reason I don't think that is we are not supplementing here with any free proteins at all. So if the proteins themselves start to fall off the DNA fragment that they're bound to, it's just going to fall apart, diffuse out into the fluid. So I think it's just the whole thing is somehow releasing from the DNA. It may be that just the contact with RAD54 in the DNA are released, and you still maintain some contact with RAD51 so that this whole thing can kind of flop around a little bit. We just don't know what happens during that switch. Yeah, I need to emphasize, we do not know, so that's just all speculative. But it's not just completely falling apart. Yeah. Typically, so those distances are difficult to measure in vivo because those are transient intermediates. And so a couple of people have made the measurements, have tried to make those measurements, generally by sort of PCR-based things where you're looking for just a single-stranded DNA overhang length. They tend to be on the order of hundreds to low thousands. So they're not huge. They tend to be relatively small. And you can, in fact, as you know, same we've shown, but other people have shown it, you can drive recombination with even smaller overhangs. And it's thought that you can do that even in vivo when you design experiments such that only a small fragment of the overhang can bind to its target. Yeah. So if I understand you correctly, the question is, how is the structure of the DNA in the cell, how might it influence all of this?
Right, in case of modified.
Yeah, so modified DNA, or I mean, you can think of it on multiple levels. Modified DNA, covalently modified DNA, nucleosomes on the DNA, DNAs with varying degrees of supercoiling. We do think all of that is going to, we know all of that is going to influence this reaction inside of the cell. How it's going to influence the particular aspects that we're able to measure, I do not know. I think if you have highly negatively supercoiled DNA as you expect, it's probably easier for RAD54 to unwind it, and so it would make the search more efficient in that case. The bigger question in my mind is, how do nucleosomes and chromatin structure influence all of that? Right now we do not know the answer to that. We know that we can take our 21 nucleotide overhang that goes 150 base pairs per second, and we can crash it into single nucleosomes. And it will do a couple of different things. It will either just kick the nucleosome off the DNA, sometimes it goes past it, but sometimes it gets stuck. What we have not done is taken that 1,000 nucleotide overhang that goes 800 base pairs per second and ask, what does it do when it hits a nucleosome? I tend to think it's going to have a little bit of a better time getting through them. But then this is also, there's also a caveat in this, in that I'm only talking about two proteins, RAD51 and RAD54, and nucleosomes too, so add a couple more proteins. In the case of in vivo, you have to have 20, 30, 40 factors that are involved. Some of those are chromatin remodeling proteins, and so we don't think that this is happening in isolation. Probably what's happening is this is coming in, and there's nucleosome remodelers that are also involved and help to move things out of the way. But again, those are kind of the edge of what we might know.
Correct versus incorrect orientation thing.
Yes.
So if I understand correctly, you're saying that the RPA helps fold the DNA so that you can loop in the incorrect orientation. So if that is the case,
Yes.
then one thing is that I would expect for longer DNA, when you had these longer overhangs, I would have more flexibility, and therefore I would see more of these incorrect things as opposed to...
So this is a super interesting question, and that's exactly what we thought. So let me make sure I'm clear. We do have a model where RPA is folding that DNA to give you both, so that you can recognize both strands with the correct polarity, one of which is correct on its own, one of which is correct by virtue of the RPA interaction. When we were making those longer and longer overhangs, we thought, okay, this should change, first of all, the amount of RPA that's there and how it's recognizing correct versus incorrect. When we went from 21 to 90 to 150, we saw the same number of RPA molecules bound, 3. Okay, so we actually don't think we're changing the amount of DNA that's being opened. It was only when we got up to 1,000 that we went from like 3 to, I can't remember the number, 6 or 10 or something like that. And the issue there is that we may actually have, if you think about 1,000 nucleotide stretch, we may actually have multiple, like instead of opening one place, we're opening in 2. But at this stage, we actually think, irrespective of length, you're opening about the same amount of DNA. That's why we got this sort of cap up to 150 base nucleotides of just 3 molecules of RPA. That's why we think adding longer and longer DNA fragments doesn't change that first passage recognition efficiency. It caps right off at 30% for all the molecules that we look at.
And there's no change in the lifetimes depending on the correct versus incorrect orientation.
So lifetime after you've recognized the target.
Yes.
We have not made that measurement rigorously. There was no obvious change. Essentially, what that means is within the time windows of our measurement, once you're at the correct place, you tend to stay there. We didn't see them
Because it's
coming
still
off.
competing with the bending energy of the DNA fragment, right?
So I don't think so, because if you recognize from the incorrect and you start driving pairing, you probably just flip around to the correct orientation. Right? Once you start driving pairing, you're going to pair across the whole thing. I think.
Hi. So I wanted to ask, with like RPA bound to the ssDNA, shouldn't the binding of RAD51 be affected and consequently the RAD54? And also how the dynamics will change in the presence of proteins like RAD55, 57, and 52?
Yeah, great question. Great questions. So let me address the first part. The second part I think is fairly straightforward. So generally when people think about RPA binding, I talked about how the DNA end is processed to make a 3' overhang, and what happens is RPA is the first protein that gets there. And so that's a key step that RPA is needed at. And then what has to happen is RAD51 has to displace that RPA. And so when you think about BRCA2 helps RAD51 displace that RPA, and when you don't have BRCA2, you can't make the RAD51 filaments. But in our case, what we're actually doing is pre-assembling those RAD51 filaments. So we're bypassing that step entirely in this set of experiments. And so RPA is not getting onto our little DNA fragment with the overhang. We think it's only binding to that open, what we think is an open gap on the double-stranded DNA. And in fact, if we just omit that little fragment entirely, we still see RPA targeted to RAD54 and tracking with it along the double-stranded DNA. Now, the second part of your question is related to that, and that is how do things like RAD55, 57, RAD52, and all the other flavors of RADs, how do they affect this? Essentially, those are affecting the reaction at an earlier step, right? So what they do, all of those guys that you mentioned, what they do is help RAD51 to assemble onto the single-stranded DNA. We actually do lots of papers, lots of studies on that assembly process, but in this case, we don't do it. I don't think they would affect because we're bypassing all of that entirely. I don't rule out the possibility that they might affect it, but at least from what we know so far about how those things work, I'm not sure that they would affect it. For RAD55, 57, there's no reason to think that it actually binds to the RAD51 filament after it's assembled. We can show that it promotes assembly, but then it falls off entirely. RAD52, again, I tend to think it's involved in an earlier step in the reaction. I'll add the caveat, we have not done that experiment, so maybe I'm wrong, but I don't really necessarily think it would influence this part of the reaction. The way most of these DNA motors are thought to work is they're actually interacting with the phosphate backbone. And so if you think about actin or a microtubule, you have these repeating units that have a defined polarity so that your adenine goes one way and your kinesin goes the other way. In this case, all the repeating units are going to be the phosphate groups in the DNA, which also have, in a sense, a plus and minus polarity or a 5' and 3' polarity. So we think that they're grabbing onto that and sort of stepping along that backbone as they go. The way they work, well, I did this, but I'm going to do something simpler. They have this sort of clamshell shape. ATP hydrolysis and binding drives this opening enclosure. The DNA would be bound across this, and it's sort of stepping as it goes along the DNA.
When this complex is bound to the DNA and it's looking for its target sequence,
Yes.
That's where you're showing that it's moving in one direction.
Yes.
But then essentially you have the zigzag motion.
Yes, yes.
In case it misses the sequence and it has to come back.
Yes.
This complex, does it exactly have a way to understand that it's missed the sequence?
No. Well, that's an interesting way to think about the problem. If it hits the sequence, it stops dead in its tracks. Even if it was going 800 base pairs per second, if it hits the right sequence, it stops. And so in that case, it knows that it's done the right thing. We don't know, I have no reason to think if it goes past the target sequence, that it knows it went past the target sequence. I don't know that the target sequence modifies it in a way that would allow some sort of molecular memory effect to say, hey, wait a minute, I missed it, I just turned around. I think that turning behavior is purely stochastic. And the frequency of turning that we see just supports that idea. Yeah.
Eric, I wanted to ask something kind of related to what he's asking.
Yes.
About this missing thing, right? So if you're thinking that it is opening up and that is where the recognition is happening, right?
Yeah.
Can you artificially have a couple of such bubbles or openings in there and then
That's a great experiment. The idea would be to pre-open
Yeah.
the DNA and just see does
whether
it recognize it better depending on how much you have
Yes,
open.
yes.
We've not done that measurement.
Okay.
That's a
That's
great
a
experiment.
really interesting
Yeah,
one.
great experiment.
Understand. So you were showing those chymographs. So you
Yes.
said that this RAT54, it
Yes.
has a switching rate where it can turn direction and move, like
Yes,
which will
yes,
give
yes.
you this zigzag in
Yes,
the chymograph.
yes, yes.
But
I think that's probably just its dissociation rate constant on a microscopic level.
But most of the movies, it seemed that they had the positive slope until it became constant, where they reached the target. So probably the switching rate is very low, because
It is relatively low.
because if it was
relatively
high,
low.
one would see a lot of zigzag before it
Yeah,
reaches
yeah,
the
no,
target.
no, no. It is relatively low. That's a relatively frequent event. Like, as I mentioned, typically, so you're going 150 base pairs per second for our 21 nucleotide guy.
Yeah.
You're going 5 kilobases, 5,000 nucleotides before you ever turn around. When I talk to audiences that work on DNA repair, this PUC19, this plasmid-based assay I showed you, that's a very typical assay. And I point out, 5 kb is the size of that plasmid that you're using in your assay. So when you put this thing in there, by the time it hits that, it's going to go all the way around it, right? And so it goes quite far distance before that turning it happens.
So again, as you could all see, how elegantly Eric can share some of the very, very complicated stuff and tries to simplify it for us and even for himself, perhaps.
Yes.
But again, it's absolutely a great pleasure to have him here for this Institute lecture. And also, he is our distinguished visiting professor in this institute. I forgot to mention that. So hopefully we will hear more about his research in the next coming years, maybe once or twice. Thanks so much for coming, Eric.
Thank you.
I'd request on behalf of the Institute, Chair of our department to please give this memento to Eric.
Thank you, Richard.